Hello students, welcome to EPG Pathshala. I am Dr. Jasjeet Kaur Narang from Khalsa College of Pharmacy, Khalsa University, Amritsar. Today we are going to discuss about a module, Basics in Cosmetics, under the paper Cosmetics. The objectives of the present presentation and the module are to study about number one, the basic liquid preparations, which are biphasic in nature. These include emulsions. We are going to study about their introduction, the types of the emulsion, the identification tests involved for these emulsion preparations, preparation of these emulsions, as well as the stability of emulsions. The second topic covered would be suspensions. Under this, we are going to study the selection of the wetting agents, the suspending agents, dispersing agents, as well as we are going to study about the preparation of suspensions, as well as the stability of suspensions will be looked into. The third topic covered during this module will be creams. An introduction about the creams, its different types, as well as the stability of these biphasic formulations and creams would be discussed. Lotions are also a part of the module. A brief introduction with the different types of lotions, preparation of lotions, as well as the stability aspects of lotions are going to be looked into. First is the introduction. Nowadays, aging of the skin is one of the most common problems in the human population. Both intrinsic molecules, example the reactive oxygen species, also commonly referred to as ROS, as well as the environmental factors like the ultraviolet radiation, are responsible for the alteration in the skin appearance and the functions. Wrinkles, uneven pigmentation, and skin atrophy are indicative of the individual's aging. Due to all these changes which are occurring, an individual also suffers a decrease in the social comfort as well as the self-confidence. In the manufacturing market of skincare products, topical applications in the form of emulsions, suspensions, lotions, and creams, the most common being the sunscreens, which help in rejuvenation of the aging skin, along with these antioxidant properties which retard the aging, are gaining importance. Biphasic liquid formulations are discussed in this slide. These biphasic liquid preparations are composed of two phases, which can either be both liquids, resulting in the formation of emulsions, or one phase can be a solid phase dispersed in a second liquid phase, thereby resulting in the formation of suspensions. First, we discuss emulsions. Emulsions are widely used in the field of cosmetics. An emulsion is a dispersion or a thermodynamically unstable biphasic liquid preparation consisting of at least two immiscible liquids. in which the dispersed phase is composed of very small globules of liquid distributed in a second liquid phase, the whole system being stabilized by a third substance known as an emulsifying agent. The function of an emulsifying agent is to form a film around the globules in order to scatter them indefinitely in the continuous phase so that a stable emulsion is formed. An emulsion system mainly consists of two phases. First, a dispersed liquid, which is also known as an internal or a discontinuous phase, and a second system known as a dispersion media, also commonly referred to as an external or a continuous phase. Stabilization of such a system is the main target in formulating emulsions because all the emulsions suffer from the problem of phase separation. The selection of the oils and the emulsifiers determines the product formed, its appearance, and the viscosity. The different types of emulsions are given in table 1 below. First type is oil and water type emulsion, which has a droplet size from 0.2 to 50 mm. The characteristics are that in this kind of an emulsion, the dispersed phase is oil and the continuous phase is water. The second type of emulsion is a water and oil type emulsion. The droplet size of such an emulsion ranges from 0.01 to 0.2 mm. The dispersed phase in such an emulsion is water and the continuous phase comprises the oil. The diagram shown on the right side clearly depicts both the type of emulsions, the oil and water and the water and oil, the difference being which phase represents the continuous phase is different in both the cases. Third type of emulsion is the multiple emulsion, which can be of two types. It can either be oil in water in oil emulsion or water in oil in water emulsion. The characteristics of multiple emulsions are that they are complex polydispersed systems in which both the oil in water and the water in oil emulsions exist simultaneously, the system being stabilized by a lipophilic and a hydrophilic surfactant respectively. The fourth type of emulsions are the microemulsions. They are thermodynamically biphasic stable systems which are optically transparent and are stabilized by a surfactant and a co-surfactant like sodium lauryl sulfate or SLS. A microemulsion is typically depicted in the diagram given on the right side, where we have a lipophilic part of a surfactant, the hydrophilic part facing the hydrophilic phase, and the whole system is being stabilized by the surfactant. Identification tests. We need to identify the types of emulsions we are using, and in order to facilitate this, Table 2 depicts the various identification tests which are used to identify the emulsions. The first test of identification is a miscibility test. In this test, both the emulsions are diluted with water. In the case of an oil and water emulsion, the emulsion diluted with water will be miscible with water, but not with the oil. However, in the case of a water and oil emulsion, the emulsion will be immiscible with the added water, however, would be miscible with oil which is added. The second test is a dye test. In this test, we add a scarlet dye to the emulsion being evaluated. In the case of an oil and water emulsion, the colored globules are seen on a colorless background. In the case of a water and oil emulsion, the colorless globules are seen against a colored background. The third test is a conductivity test. In this test, the emulsion is taken and a pair of electrodes is connected to the battery and dipped in both the emulsions through a low voltage bulb. In the case of an oil and water emulsion, the bulb glows and we come to know that the emulsion is a oil and water type because water is in the continuous phase and is a good conductor of electricity. In the case of a water and oil emulsion, the bulb does not glow, which clearly is because there is oil in the continuous phase, which is not a good conductor of electricity. The fourth test of identification is the fluorescence test. Certain oils exhibit a physical property of fluorescing in the presence of the ultraviolet radiation after a microscopic evaluation of the emulsion under UV radiation. In case the droplet's fluorescence, when seen microscopically, it indicates that oil is present in the dispersed phase and therefore it can be concluded that the emulsion is an oil in water type of emulsion. If the whole field fluoresces, it clearly indicates that oil is present as a continuous phase and the emulsion is a water in oil type of emulsion. The fifth and the last test of identification is a cobalt chloride test. In this test, a filter paper is dipped in a cobalt chloride solution. In case of oil and water emulsion, it results in the change of the color of the paper dipped in cobalt chloride from blue to pink. However, if the filter paper is dipped in a water and oil emulsion, no such change in color is observed. This slide indicates the methods of preparation of emulsions. Emulsions are prepared either by a dry gum method, number 2 by a wet gum method, number 3 by a bottle method, number 4 by using a hand homogenizer, or the simplest being the beaker method. Table 3 depicts the different methods for the preparation of emulsions. The first being the dry gum or the continental method. In this method, the oil is first triturated with gum with a little amount of water to form the primary emulsion. The trituration is continued till a characteristic clicking sound is heard and a thick white cream is formed. Once the primary emulsion is formed, the remaining quantity of water is slowly added to form the final emulsion. Number 2 is the English or the wet gum method. In this method, the proportions of oil is to water and the emulsifier used are same. That is, they are present in the ratio 4:2:1. But the order or the technique of mixing is different. In this, one part of the gum is triturated with 2 parts of water to form a mucilage. Then 4 parts of oil are added slowly in portions while triturating. After the complete addition of oil, the mixture is triturated for several minutes to form a primary emulsion. The third method is the bottle or the Forbes method. In this method is widely used to prepare emulsions of substances having very low viscosities and containing volatile oils. In this method, one part of powdered acacia or some other gum is placed in a dry bottle and 4 parts of oil are added to it. The bottle is capped and thoroughly shaken. To this, the required volume of water is added all at once, and the mixture is shaken thoroughly until the primary emulsion forms. It is important to minimize the initial amount of time the gum and the oil are mixed together. The gum will tend to imbibe the oil and will become more waterproof. The fourth method is the beaker method. This method is used to prepare the emulsion by using a synthetic or a non-gum emulsifier by dividing the components into water-soluble and oil-soluble components, which are further dissolved in their respective solvents, which can either be water or oil. Both the phases are heated to a temperature of 70 degrees centigrade using a water bath. The internal phase is then added to the external phase with stirring until the product reaches the room temperature. Mixing of such type of emulsion is carried out in beaker, mortar, or in case of creams and ointments, in the jar in which they are dispensed. Number 5 is hand homogenizer. It is hand-operated equipment in which the coarse emulsion is passed through a fine orifice. The preparation is placed in a hopper and the up and down movement of the handle of the homogenizer causes the emulsion to draw through an inlet and pass through the orifice to get dispersed droplet size of about 5 microns or less. Example is Silverson mixer homogenizer, which is widely used for this purpose. In this slide, we are going to talk about the stability of emulsions. A stable emulsion is the one in which the dispersed phase retains its initial character and remains uniformly distributed throughout the continuous phase during its storage, besides being free from bacterial growth during its shelf life. The various types of deviations from such an ideal stable behavior of emulsions results in instability in the emulsions. The commonly encountered instabilities in emulsions are: number one, cracking. Cracking of an emulsion system is due to the coalescence of dispersed phase globules, which are difficult to redisperse by shaking. This is due to the presence of microorganisms or can be due to a change in the temperature or can also be attributed to the decomposition of the emulsifying agent. The second most commonly encountered instability is creaming. Creaming results in separation of an emulsion into two regions in which there is an upward movement of the dispersed globules to form a thick layer at the surface of the emulsion. Creaming of an emulsion should be avoided because it may lead to the third type of instability, cracking, along with a complete separation of the two phases. The next type of instability occurring in an emulsion is phase separation. In phase separation, there is a change of one type of emulsion into another type. That is, an oil and water type of emulsion can change to a water and oil type of emulsion and vice versa. The reasons attributed for phase inversion include a change in the temperature, or it can be due to a change in the emulsifying agent. This slide depicts pictorially the different instability problems occurring in the emulsion. We can clearly see that how a stable oil in water emulsion looks. However, in case of any instability, which can be either coalescence, creaming, or phase inversion, the stable oil in water emulsion clearly becomes unstable and unfit for use. In coalescence, the different dispersed phase droplets join together, or coalescence is seen. In creaming, the dispersed phase accumulates either at the top or at the bottom depending on the density. In the case of a phase inversion, we can clearly see that an oil and water initial emulsion is changed to a water and oil, and there is a swapping of the dispersed phase and the dispersion media. Now we discuss the suspensions. Suspensions are heterogeneous mixtures in which the solid particles, which comprise the internal phase or also known as the dispersed phase, are dispersed throughout the fluid known as the external phase or the dispersion media by means of a mechanical agitation and use of various excipients or suspending agents. The size of the solid particles which are dispersed ranges from 0.5 to 5 microns. The particle size is a very important factor for the formulation of suspensions. In case of an external preparation, the suspensions meant for the skincare purpose should have a very small particle size in order to avoid any gritty feeling to the skin after application and to cover a greater area of application. For example, lotions containing the suspended particles evaporate when applied to the skin, leaving a very light deposit of the medicament on the surface. Calamine lotion is a suspension type preparation which is applied on the skin to provide a protective effect. The suspension should be free from any harmful microorganisms as well. Table number 4 depicts a difference between the flocculated suspensions and deflocculated suspensions. In the case of flocculated suspensions, the particles form a network-like structure due to loose aggregates, whereas in a deflocculated suspension, the particles exist as separate entities. The rate of sedimentation is high in the case of flocculated suspension, whereas it is low in the case of deflocculated suspension. Sediment is rapidly formed and it is easy to redisperse in the case of a flocculated suspension, whereas the sediment is slowly formed and it is very difficult to redisperse in the case of a deflocculated suspension. In a flocculated suspension, the supernatant liquid is clear, whereas it is not clear in the case of a deflocculated suspension. In a flocculated suspension, sediment is loosely packed and does not form a hard cake, whereas a hard cake is seen in the case of a deflocculated suspension. The floccules stick to the side of the bottle in the case of a flocculated suspension, whereas no such sticking is observed in the case of a deflocculated suspension. Because of all the above factors, the flocculated suspension is non-pleasing in appearance, whereas a deflocculated suspension is pleasing in its appearance. Also, the bioavailability of a flocculated suspension is much higher as compared to a deflocculated suspension. This slide clearly helps you to identify a flocculated suspension and a deflocculated suspension. While selecting a wetting, suspending, and the dispersing agent, we have to be very careful. Wetting agents are surfactants that lower the interfacial tension as well as the contact angle between the solid particles and the liquid vehicle. Their concentration ranges from 0.05 to 0.5%, and the rate of the wetting is often determined by placing a measured amount of the powder on an undisturbed surface of water containing a given concentration of the surfactant and measuring the time required to completely wet and sink the powder. Suspending agents are a very important part of the suspensions. These are lipophilic or can be hydrophilic colloids, resulting in the formation of colloidal dispersion with water, resulting in an increase in the viscosity of the continuous phase. They act by forming film around the particles, thereby causing a decrease in the interparticle attraction. Suspending agents help the active pharmaceutical ingredients to stay suspended in the formulation and prevent any kind of caking at the bottom of the container. A well-formulated suspension with a good suspending agent should be able to easily suspend by the use of moderate agitation or shaking. An ideal suspending agent should have the following characteristics. Number 1, it should produce a high viscosity at a very negligible shear. that is, during the shelf storage, and it should result in a very low viscosity at a high shear rate. That is, it should be free flowing during the agitation, pouring, and spreading on the skin. Pseudoplastic substances, examples of which include tragacanth, sodium alginate, and sodium carboxymethyl cellulose or SCMC, show these desirable qualities and produce shear thinning systems in which, when shaken or agitated, the viscosity diminishes. A suspending agent, that is thixotropic as well as pseudoplastic, proves to be very useful since it forms a gel on standing and becomes a fluid when disturbed. Examples include bentonite and carboxymethyl cellulose, having both the pseudoplastic and thixotropic behavior. Suspending agents should coat the particles and thus make them very less prone to caking as compared to the uncoated particles. Examples of the suspending agents which are used for the formulation of suspensions include alginates, carboxymethyl cellulose and its derivatives, microcrystalline cellulose, acacia, tragacanth, xanthan gum, bentonite, and carbomuls. A dispersing agent. An agent either surface active or non-surface active polymer which is added to a suspension or a colloid for the purpose of preventing the settling or clumping and to improve the separation of particles is known as a dispersing agent. The process of dispersing proceeds with three basic steps. In the first step, there is a wetting of pigment agglomerates. Second step involves the de-agglomeration of pigment agglomerates. And thirdly, there is a stabilization of the resulting dispersion against flocculation. This slide depicts the method of preparation of suspension. The first step is size reduction, which can be achieved by using different equipments like ball mill, hammer mill, etc. The second step is that the pharmaceutical powder with the appropriate particle size is wetted with some amount of the water miscible solvent. The third step is that after wetting, a suitable suspending agent with other ingredients is added to a vehicle and added to the above solution to form a slurry. The slurry is then transferred to a measuring cylinder and the volume of the dispersion made up. This slide depicts the stability of emulsions. We can assess the stability of suspensions by a number of techniques. First, by assessing the sedimentation volume. Second, by assessing the rheological properties using suitable equipments like Brookfield viscometer. Third, by assessing the micromeretic parameters of the suspensions. And last, the stability can be assessed by assessing the electrokinetic parameter like determination of zeta potential or the surface charge. Creams. Creams are the skincare products that are intended to be either rubbed or to be applied to the human body or any other part for cleansing, beautifying, promoting the attractiveness or for altering the appearance. These have a unique composition that can alter the ability of ingredients to reach the skin surface, which also influences the product aesthetics. Creams can be also divided as oil in water or water in oil. Depending on the main ingredients and the purpose, they can be further classified as makeup creams, cleansing creams, night creams, winter creams, all-purpose creams, and creams for the dry skin. The different types of creams and their description is given in the slide. The stability of biphasic liquid formulations and creams has already been covered under the heading Stability of Emulsions. Lotions. These are low viscosity preparations which are applied to the unbroken skin with bare hands or a brush or a clean cloth, etc. These are intended to provide smoothness to the skin, to moisturize it, and to soften it. The different types of lotions can either be bath lotion, baby lotion, hand lotion, or the body lotion. The different types of lotions are described in this slide. Bath lotion is applied after taking a shower. Body lotion, it is essential to be applied daily to keep the skin looking fresh and healthier. Baby lotion, as indicated, they are intended for the sensitive skin as they are dermatologically approved. Hand lotions, they are intended to prevent and save the hands of the individuals, especially when the hands are coming in contact with harsh chemicals and other substances leading to their dryness and dehydration. This slide indicates the method of preparation of lotions. It can be prepared by having an aqueous phase, an oily phase, the system being stabilized by the addition of an emulgent. Besides this, we can also include a majority and a variety of ingredients like antioxidants, preservatives, fragrances, and dyes, which improve the organoleptic properties as well as the aesthetic feel of the product. The stability of lotions has also been discussed under the stability of emulsions and suspensions. Their stability is also same as emulsion and suspension because lotions are also biphasic system. So students, at the end of the module, let us summarize what we have learned in this module. For the cosmetic skincare products, emulsions and suspensions are the best delivery systems as they offer both therapeutic as well as the cosmetic uses. Therapeutically, they are the carriers for the delivery of drug at the site of action. In case of cosmetics, these have been used for the formulation of various cosmetic products like creams, cold creams, liniments, lotions of various categories, etc. Various emulsifying agents and suspending agents are used for emulsion and suspension formulations in order to stabilize these formulations for longer periods of use and time. These formulations help in maintaining the skin's natural integrity and protect the skin from various diseases when used therapeutically. They are also used for beautifying purpose like foundation creams. Overall, these biphasic systems are the best vehicles for the skin care. Thank you.